yeah, this is how the area of the distribution of the plot. 58 different tree species was identified. Basically, I could say that the 60% of these trees are Cryptocephalum, Erythrophilium, Burki africana, Dicostraxis, and Jubernadia, but I call your attention for Cryptocephalum. And it's mainly 20 species they made, 90% of the area that were measured. So going to understand how the plots are disturbed, let me say that we found this pattern here in the plot, and we said, okay, there is Cryptocephalum here, there is this area, and we could say, is it a dry forest? Why? We say that because it is dominated by Cryptocephalum. Of course, we have Brachystegia jubernadia, but they are not the main dominated. There is no fire in the area. It is just mosses and grass, high relative density, and the people also use the area for different purposes. This area looks like this. So if you go in Africa, we talk about dry forest. We have only dry forests classified appearing in Zambia, but we could say, okay, you guys have only dry forest in Zambia already? Yes, okay, let's see about compare the plots that was done in Zambia and with the plots that was done in Angola, and this is the result that we found. So basically, this is how similar could be the plots in Zambia and in Angola. So just to finish, so we can say that the different vegetation types we can have in Angola, not only miombo. We can say also that it is important to classify very well the savannas, and according to the different drivers, fire mainly. And also we can say that the local knowledge of the communities that lives in this area, it is very important, because sometimes we as a scientist, when we go in an area, we don't consult people that lives there. We do with our knowledge and we think we know everything, but actually we are discovering something that people that were living there know since a long time. Thank you.
Thanks to Lima for a very interesting talk. I must say it's also really nice to see work coming from these parts of Africa that have previously been neglected at this meeting, so that's really nice. Next up, Carl, over to you, a 15-minute talk.
Thanks a lot, Ben. Yeah, the key point on that was that if you look at the maps of dry forest of Africa, they're only apparently in Zambia, but then they clearly occur in Angola, and the people there knew it all the time. So the talk I'm going to give has a rather grandiose title, apologies, but this is sort of my goal over the last 6 to 10 years, and it's my goal for the next 10 years as well, probably. So if you're interested in this goal, come talk to me. And the talk you're going to get now is just one little chapter in my journey on that goal. So, and I'm going to first talk about the perspective on the dry tropics from the Americas, and specifically about tropical dry forests. So, people who work in the Americas tend to divide the dry vegetation into savanna and dry forest. And the dry forest can be things that look very foresty, with a tall, closed canopy. Usually deciduous in the dry season is this place here. But then what we call tropical dry forest can also have an open canopy and a rather short canopy. It can also have a thick, short, closed canopy, a bit like what people here call a thicket. And this dry forest has a very patchy distribution across the tropical Americas. So this is just lowland tropical South America east of Brazil, where I worked for a long time before moving to work more in Africa. And we see that these dry forests occur in little patches across the region. But one thing that is quite notable about these tropical dry forests in the Americas is that they show strong floristic coherence. So this is an analysis from one of my PhD students, Pedro Silva Miranda, and each one of the points at the end of this plot would be in a place in space where we have a list of the tree species present at that site. And so this is over 4,000 sites from the previous map where we have some idea of what tree species are present. And then you can do a clustering analysis like you saw in the last talk, where you group sites based on their similarity and tree species composition. And this is the output here. So savannas form one clear group in this analysis, savanna sites, and dry forests form another clear group. And then the moist forests fall into two different groups. Chaco, which has temperate lineages, falls somewhere else. But anyway, so despite all of that heterogeneity in what that vegetation looks like, from a tall closed canopy forest to a more open scrubby vegetation, it's all floristically falling together. The same tree species are dominating that. And so another thing, sorry, I should have mentioned, that distinguishes these tropical dry forests from savannas is the adaptations of the taxa they're in for fire. So the savannas have a lot of C4 grasses in them. They burn regularly. The trees are used to dealing with fire. And the dry forests, most of the taxa are fire sensitive. There's a lot of succulents in tropical American dry forests, like cacti. And so this fortuitous coincidence, or perhaps fortuitous, perhaps not, between the floristics and the way these ecosystems function, make us feel quite confident about asserting that the tropical dry forests in the Americas are a distinct biome from savannas or moist forest. Right? So that's my understanding of the world. And then I started to work in Africa, more so in 2017, and things kind of fell apart. So I'm going to give the African perspective on the tropics from someone who's not from Africa and has only worked here deeply for the last 6-7 years. And please, after the talk, come tell me what I got wrong. So if we looked back to, this is one classic paper on dry African vegetation. The ideas go back further than that, but Huntley summarized them nicely in this book chapter. And he divided the savannas of Africa into the arid savannas and the mesic savannas. These arid savannas are the fine leaf savannas or the fertile savannas, and these mesic savannas are the infertile savannas where there's lots of bleaching from rainfall, and the broadleaf savannas. And there's just a couple pics from places where I work. And so if you zoom out a bit and you look broadly across Africa, so you have savannas, and then when you look for the forests, where are the dry forests of Africa? So this is, I think many scientists here divide at the first division, the vegetation of Africa, in the tropics at least, between forest and savanna. But there's nothing dry forest in here. This is mostly all moist forest. So where are the dry forests of Africa on the maps? Maybe that little bit of cryptocephalum forest in Zambia that spills into Angola. But some other candidates to think about. So what about thickets? So I said a lot of the vegetation in the Americas is thickety in form and has succulents. There's succulents popping up here and there in this vegetation. So maybe thickets are the dry forests of Africa. Or what about the forêt dents sèches of the Francophone research world? Sorry, that flipped sideways. I don't know why. Trees don't usually grow that way, but it's supposed to be going up. That's a pig Entendophragma. And so if you read the Belgian French literature, they point towards this forêt dents sèches, which is quite often in a matrix of mesic savannas, but you have these patches of dense forest where fire doesn't occur. Whether those patches are relics or whether they are just unique edaphic conditions is actually an open question. So I thought, okay, let's get some of these dry forest possibilities into databases of tree species composition across Africa like we've done in the tropical Americas. So this is the Afrotrop tree database, spearheaded by Adeline Féol in Belgium, but now France. And so that was in that map earlier from the Allemande paper, where she's pulled together tree species inventories, just like we have in the neotropics, for what she calls either forest or savanna across most of tropical Africa. And then working with my co-authors, who I should have acknowledged at the beginning, Susie Vetter, Sally Archibald, Kath Smart, and Vera Dekar, we pulled together additional data from other parts, other vegetation types and other parts of Africa. So trying to fill out this subtropical portion of Africa, trying to get thicket data where we could. That point should be in Zambia, it's an error. So we have thicket data from Ghana, from the Rift Valley, a bit from Tanzania, a lot from South Africa, and also some of the drier forests. So I've split the dry forests as having less than 1,200 mils of rainfall and more than 300 millimeters of climatic water deficit during the dry season. And then I did the same sort of analyses that Pedro and I did in South America, some big hierarchical clustering analysis. And I'll just go through that quickly. trying to make up time for you, Ben, if I can. Yeah. So in this first group here, we have a bunch of sites that are called shrublands in Namibia and maybe a bit of savanna, right? So these shrublands burn, by the way, so these are pyric shrublands. Then the next group down is most of the South African thicket, which is forming a clear group in the clustering analysis with a few other things thrown in. The next group down is this is what I'm calling the extratropical or subtropical mix. So it's a lot of the savannas in South Africa, maybe a few up here, and then also a bit of moist forest, dry forest, and thicket thrown in. So that's kind of a heterogeneous group that we don't totally understand. And then the fourth group is tropical forests, and so that's all the forests of the Congo and Guinean region. And the drier forests in the Dahomey Gap and transitioning to the Sahel seem to fall within this group. And also the 5 thicket sites we have from Ghana are falling in this group here. And then there's the tropical savanna group, which includes the rest of the tropical thickets, all the coastal forest in Tanzania and Kenya, which has some interesting floristic relations with the Congo rainforest, but overall floristically it's closer to savanna. There's a lot of degradation there in savanna incursion, so it's a bit like messy there. And then also, so those cryptosepalum dry forests that Louisa works in here fall within this group as well, right? So those are your floristic groups. And so far what I was reading into this is that, so the thicket and the dry forest, right, are candidates for seasonally dry, non-burning vegetation, this thing that applies, that we call tropical dry forest in the Americas, even if it doesn't always look like a forest, right? But it seems to be falling in many different places in this kind of floristic analysis. So you can use other approaches like ordinations. Clustering does have its issues. And so these are the floristic groups on the first 2 axes of a detrended correspondence analysis. And we see that even in this like ordination, those groups are pretty coherent, at least on the first 2 dimensions. And there's this clear line between the tropical vegetation, the tropical savannas and forests, and the extratropical or subtropical vegetation over here. If we break that down by vegetation type, we see that within the tropics, so this zone here, the thickets and dry forests are kind of falling near each other, so there's some signal of floristic coherence here, but not a super strong one. So this is like the first look at the analyses. We finally finished cleaning the taxonomy on this database just a few weeks ago. So just to, again, try to save Ben some time. So tropical dry forests and thickets in the Americas form this nice cohesive floristic group that correlates with ecosystem function. When we try to pull together similar databases for Africa, after a forest order segregation between tropical and non-tropical, there is some signal of floristic cohesion in Africa, but I would say it's, so I did some deeper digging after I submitted the talk, because we had to submit the talk in advance, and I'm less confident on this now. It's still a work in progress. But it's not there. So is there a general division between burning and non-burning tree floors in the dry tropics? The answer is, I don't know yet. I'm still working on it. So thanks a lot for your time.
Thanks, Carl. Wow, you did record time there. So it leaves us some time for questions. I'm sure there's a number of burning questions out there. Yeah, we got one over here.
Yeah, thank you for a very nice talk. Maybe I missed this, but where would you place the Miombo now in this?
So the Miombo is in the savanna there, as far as like a mesic savanna, but it's still, so my savanna categorization there is for vegetation that has trees, that burns regularly, has a grassy understory, etc. Yep. So Miombo would be in the savanna group. Yep. Most woodlands would be in there, most African woodlands. Yep. Rob.
Yeah, so I was sorry, I was just wondering if you took like a trait-based classification instead of a sort of a phylogenetic and taxonomic one, would that clarify
I missed a key word.
A
What
trait,
kind of-
trait
trait-based.
based.
Yeah, sure. So that could be a next step. That's a nice next step. I have a postdoc, Jens Ringelberg, who worked on the succulent biome. Some might know of him. So he's pulling together trait data that's associated with the tree species in our species list, and he's going to be looking at that for evidence of functional biomes in there. Yeah.
Okay, any more questions? Not. Okay, thanks very much, Carl. Just to point out, sorry, I missed earlier on the program, there's quite a few posters in the session, so please do be sure to come grab a glass of wine and some snacks this evening and look at these interesting posters from the session. Next up, we have Mukta, who's joining us all the way from Bangalore, from Mahesh Sankaran's lab, and you've got a 5-minute talk. Over to you.
Hi, my name is Mukta and I'm a PhD student in NCBS Bangalore. And today I will be talking about the distribution patterns of grass species in the Indian subcontinent. So we all here know that the tropical grassy biomes are ecologically and economically important, covering almost 20% of the land surface. And even in India, these biomes are widespread. The yellow portion here in the map shows the semi-arid grassy biomes, which cover around 10% of India's surface area. And now grasses form the understory in these systems, and it is obviously important to study the distribution of these species. And so for this, we collated data from 31 floras, and here we can see that we see a wide variation in the total species richness across the country. So this data consisted of grass species present in each district in India, which is the administrative unit. And our final data set consisted of 1,121 species. And we analyzed their distribution with respect to the photosynthetic pathway, life history, and the subfamilies. So when we look at the richness pattern of grasses across the biogeographic zones in India, you can see that the Deccan Peninsula, which covers the most of the area, has the highest species richness, followed by the biodiversity hotspots of Western Ghats and Himalayas. But if we look at the species richness per unit area, we can see that Deccan Peninsula has the lowest richness per unit area, which might be suggestive of a higher turnover. So when we look at the pattern of the distribution with regard to photosynthetic pathway, we see that the C4 species form the major proportion in all of the biogeographic zones except for the Trans-Himalayas, and the proportion of C4 species is highest in the hot and arid zones of the desert semi-arid. And the C3 species are again, like I said, form the higher proportion in Trans-Himalayas, which is a cooler biogeographic zone. So when we look at the relationship of the species richness with climatic variables, we saw, after controlling for the district area, we saw that the C4 species richness increased with mean annual precipitation, mean annual temperature, but it decreased with precipitation seasonality. Now, as opposed to C4, we saw that the C3 species richness increased with mean annual precipitation, but it decreased with mean annual temperature and precipitation seasonality. We also looked at the proportion of C4 species, and we saw that as opposed to the C4 species richness, the proportion decreases with mean annual precipitation, increases with mean annual temperature and precipitation seasonality. Now, if we look at the distribution of annual and perennial grasses in different biogeographic zones, we see that perennial grasses form most of the proportion of the species throughout all the biogeographic zones, But the proportion of annuals is comparatively higher again in the hot and dry biogeographic zones of deserts and arid. And like we see for most of the taxa, even grasses show a pattern that most of the species are restricted in distribution, while very few species are widely distributed. Now, if we look at the richness of different subfamilies, we can see that Panicoideae, which is the most species-rich subfamily of Poaceae, which also consists of the tribe of Anthropogony, which is shown in green here, forms the major proportion in most of the biogeographic zones except Trans-Himalayas, which is followed proportionally by Chloridoideae, which is higher in the drier zones of semi-arids and deserts. The C3 subfamily of Poideae and Bambusoideae are comparatively higher in proportion in the cooler and wetter zones of northeast Himalayas and the Trans-Himalayas. So to summarize, we see that India is home to around 10% of all the grass species found across the globe, and the richness of the grass species varies widely across the biogeographic zones of India. And we also saw that C4 and perennial grasses form the major proportion of species in all the biogeographic zones, and their richness is correlated with precipitation, temperature, and seasonality, and that Panicoideae is the most species-rich family in Poaceae, and the C3 subfamilies of Poideae and Bambusoideae are found in the cooler biogeographic zones. And thank you for listening.
Thanks very much, Mukta. Unfortunately, we are out of time, so if anyone wants to ask you a question, please find Mukta over lunch or over the next few days. All right, now moving on next, we have Paul Applin. Yeah, you are going to be talking to us about grazing lawns in Kruger. Thank you.
Hello, I'm Paul. I'm from Ireland. I'm based in Ireland now. I'm a geospatial scientist predominantly. I like ecology, but I still get mildly terrified whenever I attend a true ecology meeting. So I'll try and stay with you today. I'm going to be talking about grazing lawns and remote sensing. I just want to get this out the way first. One of my editorial responsibilities is with Land Journal, and they contacted me, I didn't contact them, and offered to fund my trip here. So I felt morally obliged to do something back for them in return. So I suggested why don't we do a special issue in savannas? I haven't done a special issue on savannas before. I have worked in and around savannas for a while. So we've set one up called Observation, Monitoring and Analysis of Savanna Ecosystems in Land. I've got colleagues Elias Simeonakis in the UK and Moses Choate in CSIR as co-editors. And we're publishing in this journal. It's a pretty good journal. It covers a wide range of topics, and savannas fits very nicely, and change in savanna environments fits very nicely under here. I will be given some free papers to run this special issue. So as soon as this started developing, I thought, well, I'm planning to come to this meeting, so I'd like to make those free papers available to participants in this meeting as far as I can. I wrote this down, and then I came here, and then I saw how many there are of you out there. I'm slightly worried about this now, but I would like to prioritise where I can African colleagues and early career researchers and things like that. So please contact me if you're interested in this. I'll try and see if I can circulate information about this to the email list maybe after the conference. I'm going to cover a very wide range of content in the special issue. Okay, so that's out the way. So I'm a remote sensor principally. I'm a land cover mapper for most of my career. And I started off doing land cover mapping in Northern Europe, where it's very easy because you've got very regular agricultural landscape. Then I came to Kruger 24 years ago. It was after those floods in 2000 that I was first here. Very different game, very challenging environment to map because of the spatial heterogeneity. We've done some work on this. This is some previous work. This was led by Kwame, one of my co-authors here. Isaac, who's not here, is my other co-author on this paper. And we used a series of different machine learning approaches to try and map land cover in and around Skukuza. It's in the southern part of Kruger. So we used random forest support vector machine, CART, classification and regression tree, and multi-layer perceptron to classify a range of different land covers, including grazing lawns, which are the dark green class here. And we classified these things pretty well. High accuracies, over 90% in all cases apart from CART. So we were comfortable that we were able to get somewhere with high resolution imagery and sophisticated contemporary machine learning algorithms. Since coming here, it's coming here to Kruger over the years that the ecologists have told me what they're interested in any particular time. And it became apparent that grazing lawns kept cropping up as something that ecologists would be very interested in knowing about where they are, how they're changing. So they're, many of you will know more about this than me, they're very important aspects of a savanna ecosystem, linked to herbivory and fire, and important in nutrient cycling and just heterogeneity broadly. But we don't have that much knowledge of exactly where they are and how they change over time and why they change over time. We've got some general ideas, but nothing that specific. In that earlier paper we were starting with grazing lawns, but land cover mapping was more sort of straightforward at that time. But we did try and get our head around grazing lawn prediction. So we created probability surfaces. So on the left-hand side, we've got probability surface to identify the grazing lawn class, and then tried to optimize the identification of those areas and converted it into a binary map of grazing lawns. So we have started to do this sort of work at reasonable levels of accuracy, again, using those machine learning algorithms. And they were all not bad, apart from CART was a little bit less accurate than the others. What I really want to talk to you about today is the sort of more recent work, which we're looking to publish quite soon. And this was about a grazing lawn change over time. To monitor this, to try and get an idea of this, and then to identify maybe some of the causes in and around these sorts of processes. So there are 3 sites in Kruger, mostly in southern Kruger, I think just west of Skukuza, towards Lower Sabi, and then up in Satara. And we observed these over 3 dates. 2002 was the first one that first came out and did field work here. Then 2014 and 2019, using high resolution imagery, so Iconos, Worldview, and more recently Planet Imagery, which is a wonderful resource for people in tropical parts of the world to get free high resolution imagery. You have to pay for the other ones. But they were all supported by field campaigns. We were here, each of those occasions, doing land cover survey and vegetation measurement and things like that around the park. So we got a pretty rich data set going back quite a long time. So this is why we're very interested in doing this work, to try and really work out what's going on. And again, we used machine learning. I'm going to talk about random forest results today, just give you one example of that. And these are the results. These are the grazing lawn dynamics. This is just one site. This is site 3. There's a big artifact you can see in the middle image of a result of a burn scar, and burn scars are, there all the time in places like Kruger, and always creating difficulties when you're doing aerial comparisons over time. The grazing lawn class is purple here, so you can see that there's some patterns there, and there's some similarities broadly in the sort of shapes and distributions between 2002 and 2014. But then it goes a bit wild in 2019. Significant change, significant increase in grazing lawn. That's actually not necessarily what we're expecting, and I'll explain a little bit about that in a moment. But you can see there is something there. These things are pretty dynamic. They're not necessarily static. They're not staying stationary in one place. There's lots of processes going on in and around them. Looking at the aerial coverage of the classes specifically, interested specifically in grazing lawns, but again of just one of the sites. I'm just going to summarize all 3 sites in one slide after this. So in terms of aerial coverage, there's lots of details here, 2002, 14, 19, and then change between the different time periods. I'm just going to look at the right-hand column, the overall change for convenience between 2002 and 2019. And you can see there's significant changes going on. If we jump straight to grazing lawns, there's almost a 10% increase in grazing lawn cover over that time scale. Where's that come from? Interestingly, it's come principally from the taller grasses, the bunch grasses, that have seen a 21% drop in coverage at that time. The other significant change, few people will be surprised about, is from woody cover. So the previous body of work we did before grazing lawns was on shrub encroachment or woody encroachment, and we've done one of the papers, it's not in this presentation, but we looked at scrubbing up and what was offsetting this increase in woody cover in shrubs and in trees around Kruger. So that's increased by 10% as well. But significant increase in grazing lawns as well, and a significant drop, an unexpected drop in bunch grasses going on here. But this is just one site. There were, so we'll look into, I'll talk about the multiple sites in the next slide. In terms of interclass change, I don't know how your eyesight is. Mine is not very good anymore. I can't see what's going on down here, so I'm guessing most things. The colours here are quite subtle. They're very nice, but you might not be able to distinguish between them. There's significant things going on here. So grazing lawn that is unchanged is in a kind of light tan colour, and you can pick out some of that. Bunch grass to grazing lawn is the dominant green colour. Okay, and you can see that there's a lot of areas that were tall grasses that have now been reduced down to the short grass grazing lawn environment, especially in the second time point. The other colour that probably stands out is purple, and this is bare to grazing lawns, and principally there are probably artefacts along the River Sabi in this case, and these are artefacts. We're not suggesting there's real change going on here. So the three sites, these are the three different sites. So you can see west of Skukuza, Site 1. Site 2 is Sabi. Site 3 is Satara. So Site 2's got the Nkulu exclosures in them. We've done some work on that. That's quite useful to find out about sort of woody cover change and the resilience of trees when there aren't animals there or elephants there. And Site 3, Satara, has that grass experiment, the large grass experiment, which is also quite good. In terms of the grazing lawn class only, so looking at change, and here we'll look at change in both of the time periods, 2002 to 2014, 2014 to 2019, and then finally the whole time period. You can see that, so Site 3 is the one we've looked at, and you can see that there is change over time, and then accumulation of that over time, so 10% increase in grazing lawns. Well, now if we just look at the 2002-2014 change, the first time period, you can see that there's a fairly consistent trend going on here. Okay, so when we saw that, we were very happy, because it seemed to be working, it seemed to be consistent, we had replicates and so on. And then 2014 to 2019, a big increase in grazing lawns in the site that you've seen, but in the other ones, fairly static and maybe even slightly negative, ultimately meaning that there's significantly lower overall increase in grazing lawns between those sites and the Site 3 that I've shown you. So what's going on here? And there's always lots going on in savanna environments. Sorry, quite a lot of text on the slide. This is sort of the important stuff. So in terms of a discussion, so herbivory and fire and the interaction between them is very important here, of course. In the earlier time period, we think that's relatively representative of normal, in as far as you can get normal. We hypothesize it's the kind of cumulative action of grazers that's leading to widespread formation and persistence of grazing lawns. Okay, it's sort of what we were thinking before we saw it. If you remember, of course, in the 2014 to 2019 period, there was a major drought event, which came and interrupted lots of ecosystem processes and lots of things that were going on, with lots of spatially varying implications, lots of consequences that it's hard to predict. And reading further about this, we're still doing our work on working out all the results and what they mean and so on. Some investigators have suggested that droughts can decouple grazers and grazing lawns, meaning that after a drought, losses of grazing lawn through things like self-shading and colonization by tussock grasses means that you're going to lose grazing lawns. Okay, so this could explain the declines in those first two sites. The site we've illustrated here, Site 3, is very strongly influenced by the Sabi River running right through the middle of it. So higher moisture availability, maybe less drought stress overall. So perhaps that will explain why the association to grazers to available moisture suggests that there was more moisture there, grazers were still doing okay there, so grazing lawns were still able to persist and establish at a time of drought. These are our speculations at this stage. And just to summarize that, so grazing lawns, you can see them, you can find them. We've done a lot of field work going out there and giving you percentages over 90% in identifying these sites using contemporary image analysis approaches and using sophisticated image sources like the very high resolution spaceborne data. Grazing lawns are widespread and quite dynamic in Kruger, in southern Kruger and in savanna environments. Principal drivers, of course, are herbivory and fire and how they interact between them. But here, drought may have been demonstrating itself as an ecosystem interrupter, leading to, in a couple of cases, colonization of grazing lawns by bunch grasses. That's my final slide, just to remind you about that. Contact me, paul.applin@mic.ul.ie, but I'll go back to that to finish. Thank you.
Thanks, Paul, for a really interesting and fascinating talk. We actually have got a minute for a question or two. I see we got a bunch. So let's start with Susie.
Thank you. I was really intrigued by that slide that showed the one with the dull colors, the changes from to. And I didn't see any change from bunch grass to woody. The only change that resulted in woody was from grazing lawn to woody. I don't know if that agrees with your observations or how you might explain that.
No, it doesn't. It's a great point. It may be the colours.
No, it's from the legend. I looked at all the different ones that were listed.
I just can't, at the moment, I just can't see it to comment on that. But I can answer your question. When we 2017 scrubbing up paper, so in remote sensing, when we looked at change over time in Sabi and elsewhere, 3 sites then as well, including the exclosures, we found increase in shrubs and loss in trees, and that might well be related to elephants, as we've heard already today, and we found increase in shrubs from loss in grasses. And we had 2 classes of grasses, can I remember off the top of my head? Was it bunch grasses? It might well have been. I've just realised I haven't actually answered your question because I'm talking about woody encroachment and not changes between the grass. I'll need to look at the data, but I'd be...
Okay, Susie, you're going to find him at lunch. We're going to move on to Lisa and then Sam.
Differentiate between true grazing lawns with the grass species composition of a grazing lawn and something that's temporarily overgrazed. To what extent can you differentiate between that?
Now we know why Isaac isn't here today to help me answer these sorts of questions. I was sitting over there thinking, right, if they ask me this, I was trying to remember how we defined the classes when we were in the field. I think principally it was on the basis of height. But actually, it's not that hard to separate very low grasses from the tall ones. There isn't actually that much in between, in my experience in Kruger. In terms of species composition, I'm not sure. I'd need to get back to Kwame Andes, I think, just to take that forward. But I know why you're asking. It makes sense. I was sort of expecting this sort of query.
Thanks, Paul. One last one from Sam. Please keep it quick.
Thank you, Uta. I was just wondering, Bob mentioned this morning, when will Kruger change from a fire-driven to an animal-driven system? And perhaps some of those difficulties or those differences that you see, maybe at some patches this is busy happening in Kruger, and maybe it's a useful thing to reflect a little bit on.
So the question is, when will it change from one system to another? Can any of our ecologist friends here answer that?
Thanks.
It's beyond my pay grade.
You're going to have a busy lunch, Paul. All right, moving on. Next up on the program, we've got Tyler talking to us about measuring grass with LiDAR. Fascinating.
Hi, everybody. Thank you. And I wanted to just quickly thank the co-authors on this paper, Peter Boucher, Xenia Singh, and Andrew Davies, who have all been big help with the remote sensing aspects in particular, because I am not a remote sensor, so I won't be answering any questions on that. So the question that we had was a pretty simple one, which is, can we use high-resolution LiDAR to estimate grass biomass? And if we could, how does that compare to some of the traditional field-based methods that we use? And what sorts of opportunities for new research would it open up to be able to do this at a very large spatial scale? So I won't belabor the point about why grass biomass is important in savannas, other than to say that a lot of the methods that we use currently to measure biomass in the field were designed with this idea in mind of measuring some aspect of forage quality or forage quantity, right? And so we are looking at a large spatial scale and looking at average availability of biomass. But increasingly, what we're interested in, the sorts of questions we're interested in, have to do with variation in grass biomass at much finer scales, right? So we might care about something like microclimate regulation and how that affects the movement of small mammals through the grass layer. We might care about the spread and intensity of fire, which can be strongly influenced by very local scale patchiness in grass biomass. And so there's a need to both increase the resolution, the spatial resolution at which we can measure grass biomass, but also the spatial extent. How big of an area can we measure accurately? And can we get at some of the heterogeneity that we know exists in the grass layer broadly in savannas? And so as a quick reminder, these are sort of the standard field-based methods for estimating grass biomass. Importantly, all of these involve this first initial step, which is that they have to be parameterized with destructive harvesting, right? So you would go out to a site, you would measure the grass biomass with one of these non-destructive methods. This is a disk pasture meter, but there's also other visual point intercept methods. And then you'd harvest the grass right here, dry it, and weigh it. And you can develop scaling equations like these down here. These are all for disk pasture meters that are actually quite accurate, right? And the benefit of this then is that you spend a little bit of time up front measuring your grass biomass, and then you can quickly go through a site and take dozens or hundreds of measurements and pretty accurately scale them up to a biomass number, or kilograms per hectare or something like that. And so what we wanted to do was to see whether we could do something similar at a larger scale with high-resolution remote sensing. So there's a number of potential benefits to this approach, right? It's large, continuous coverage. This particular platform here can cover somewhere in the neighborhood of 1,000 to 1,200 hectares per day. It's very high resolution, so we could potentially estimate grass biomass at a scale at a pixel size of 10 centimeters by 10 centimeters, so quite high resolution. It has the potential to estimate biomass beneath trees. I'll talk about that a little bit later in the talk, but that's something that's particularly challenging for a lot of the field-based methods where you have to get an apparatus beneath a tree and then, in the case of the disk pasture meter, drop it. And you also get the simultaneous collection of all kinds of other geospatial data, including information, structural information about the tree layer. So there are a number of limitations that I do want to acknowledge. This is quite expensive to do. It's logistically challenging. And like all these other methods, it does still require ground truthing, right? So you're not getting away from that. You can't sort of use remote sensing as a way of getting away from ground truthing. But let's see if we can get it to work, right? So this is the sort of data that we collect with our UAV-borne LiDAR sensor. This is the Halo sensor. So what you're looking at here in the top is a top-down view of 24 hectares of Kenyan savanna. So the green, the sort of brighter, warmer colors are the tops of trees. And then the blue sort of you see in the background here is that grass layer. And I just want to point out, because this issue of trees will come up again later, this is the CLE herbivore exposure plots, which is why there's such strong variation in tree cover. But you can get a sense for just how common, or just how prevalent the trees are in parts of this system, right? And so if we're trying to measure grass biomass using LiDAR from above, those trees present quite a big problem. This is a side-on view of the same habitat. So all these trees are Acacia drepanalobium. This is about a 40-meter stretch. And so here you can see fairly high tree cover, right? But we still have dense coverage. All this blue band down here is the grass layer. So this is what we're interested in, and all the tree points up here essentially are just in the way. And so there's a number of ways to deal with this. And for the purposes of the talk today, I'm going to talk about the most brute force method. Here we're relying on, this is actually data from another site in Kenya, slightly different soil type, different plant community. But this is field collected data looking at the vertical position of different plants within the system. So you measure the height above ground of all the different plants in the system. And I've just broken this down here into herbaceous plants, which are in the green, and trees, which are in the blue. And there's a sort of convenient breakpoint at right about 1 meter, where if you were to cut this point cloud at 1 meter, you'd get primarily herbaceous vegetation left in your sort of remaining point cloud, and you'd be excluding everything above it, which is almost exclusively trees. You do still get a little bit of tree points down there in the grass layer, but that's one of the ways that we handle this problem. Okay, so if we were to take that big point cloud, cut the tree layer out, we're left with a dense point cloud like this, where we can have hundreds of thousands of points per square meter. And I've just drawn a little schematic up here of how we pull various height and other metrics out that we're going to use to predict biomass. So you can imagine if we wanted to look at this at a 1 square meter footprint, we'd have this cylinder, or this volume here, 1 square meter footprint, where we'd have, like I said, hundreds to maybe thousands of points. Each of these points is a LiDAR return that we're assuming is grass, and each has a height above ground. So from that distribution of points, you can calculate all these various percentile metrics, mean height, median height. But we can also do something a little bit more unique with this LiDAR data, which is to break down our volume into smaller voxels, so these smaller cubes, and look at something like the proportion of those voxels that are occupied. So in this particular example, we have a 1 meter squared pixel that we're dividing into 25 centimeter voxels, and we could look at how many of those have grass points in them, or the density of points in the average voxel. And then for the purposes of the talk today, I'm going to talk about field data collected at 2 different sites where we have these LiDAR metrics that we're going to use to ground truth and see how well we can predict grass biomass. So the 2 sites are Impala Research Center in Kenya, and particularly the black cotton soils at Impala, and then the Lower Sabi experimental burn plots here in Kruger. So at each of these 2 sites, we collected between 80 and 150 of these half square meter quadrats. So this is 50 centimeters on a side. We identified these with flags prior to flying with the drone and collecting LiDAR data. And then afterwards measured various metrics of height in the field, used a dispasture meter here in Kruger, and then ultimately harvested that biomass. And so using the RGB camera on the drone, we can then find the corners of these plots in the imagery, and then cut the LiDAR out and exactly match the LiDAR data to the field collected biomass data. Okay, so this, what I'm showing here on the left side is Impala, so Kenya, on the right side here is Kruger. This is all field data, right? Just to say that field methods, the standard field methods that we've used for decades still do work. So at Impala, this is a measurement of height. Height accurately predicts biomass here with an R-squared of about 0.7. At Kruger, we use the dispasture meter because there's a great scaling equation developed here at Kruger to do that. But again, a fairly strong correlation. R-squared here is 0.81. And I'll just point out that although the correlation is strong, it is sort of slightly off this 1-to-1 line that you might expect. So that is to say that here on the y-axis, this is what we collected in the field. So this is the true biomass. And this is our estimate using the dispasture meter. We'd expect those numbers to be on the 1-to-1 line, and they're slightly off. These are the same field harvested biomass data on the y-axis here, but now looking at LiDAR metrics instead of field metrics. So in both cases, the best fit line that we had was using this 98th percentile height metric, which is basically the maximum height of the vegetation. In Kenya, this scaling relationship works just as well, identically to measuring height in the field. So that's a good test of the fact that we can, in fact, measure grass height with LiDAR. And here in Kruger, again, 98th percentile height works sort of comparably well to the dispasture meter. It turns out that, not surprisingly, all these height metrics are strongly correlated with each other, and so you could pick almost any of our percentile height or mean height metrics, even these occupied voxel metrics, and get a fairly strong and consistent relationship between that LiDAR measured indicator biomass and actual field measured biomass. Also point out that, sort of consistent with our understanding of how these scaling equations work for methods like the dispasture meter, the relationships are quite different for Kenya here in the red and Kruger in the blue. Very different grass communities, very different soil types, different rainfall, and so we'd expect these relationships to be different. So the next thing we wanted to ask, so we feel like we have a fairly good ability to measure grass biomass that is sort of comparable to what we do in the field. The next question is, can we use it to infer anything about how savannas respond to disturbance, and is it a useful tool for understanding grass biomass at a larger scale? So I'm going to talk quickly through two case studies, one from Kenya, one from Kruger. And just quickly talk about some of the pros and cons of this new method. So this is the Kenya long-term exposure experiment. It is, each of these plots is a 4 hectares, so 200 meters by 200 meters, 6 different treatments that progressively exclude more of the herbivore community, and in this case, crossed with cattle. So the sort of big comparison here is a full exposure plot here compared to this one here, where you have all wildlife accessing the plot plus cattle. And you can see quite strong differences there in tree cover. So the way that historically data has been collected in CLE is using this point intercept method. So this is a 10-pin frame that's measured at each of 5 points within each plot moving forward. That was historically up to 100 points per plot. And these were parameterized several years ago using 3 harvested plots, 3 harvested subplots per plot. And so this is what the field data looks like from 2022, which is when we did these LiDAR flights in Kenya. I'll note that this was at the end of about a 2-year drought period, and so there's not a lot of difference between the plots here, but this is fairly consistent with historically what has been the case in CLE. And as you'd expect, where you have no herbivory, you've got higher grass biomass. Where you've got the most herbivory, you have the least. So if we overlay in the lighter bars our LiDAR estimates using those scaling equations with the 98th percentile height, you can see, broadly speaking, we've captured the same sorts of patterns, right? We have the highest biomass in the plots where we don't have any herbivores, and lower but sort of consistent biomass across the other treatments. I'll mention that we just selected 10 sort of haphazardly placed 10 by 10 meter plots within each of these to estimate our biomass. So pretty good agreement here, except that now that I reveal this second Y biomass, you see that the actual estimates are quite different. Right? And so our LiDAR estimates are, the average is somewhere here around 1,000 kilograms per hectare, whereas the field estimates are maybe 500 or 600, right? So a doubling in the actual biomass. We're still sort of trying to figure out what might be going on here. I think one of the potential culprits has to do with spatial scale in this case. So the CLE plots, the field data was originally collected at the 1 meter sort of quadrat level, but then ultimately pooled at the scale of the 4 hectare plots before that scaling equation was developed. Whereas our LiDAR calibration was done, like I said, in these 50 centimeter by 50 centimeter quadrats, and then ultimately we estimated it at 100 square meter subplots. We tried to sort of match and get between these 2 levels with our LiDAR, but it is possible that spatial scale plays a role here. The second example that I'll just talk through quickly is from Lower Sabi. So these are the experimental burn plots. We did the same field collection methods here. So these are what the quadrats look like with the flags in the corners. You can get a sense for that. And then similarly, we estimated LiDAR with in 10 100-meter subplots per plot here. Okay, so no big reveal here other than to say that we have a similar problem, in this case in the opposite direction. So we have pretty good agreement. I just picked out 5 of the plots here, a few that have some of the highest biomass and a few that have the lower biomass. Fairly good agreement across the plots here. There is one that has a bit of a disagreement here. This may be one of the more heterogeneous plots, and it may just be that where the dispasture meter measurements were taken and where our random subplots were didn't quite line up. But that's one potential issue. Other than that, we have strong agreement. And then, like I said, we similarly have this disagreement with the absolute magnitude, but not the sort of pattern at the plot level. Here, I think we have a slightly different issue, which is that the scaling equations that we're using to go from those dispasture meter measurements to grass biomass were developed in Kruger, but for mixed grass systems. And so we're actually using a scaling equation that may be inaccurate for that system. And so although our LiDAR estimates have a pretty strong ability to predict grass biomass, we may be losing something here by using scaling equations that were not developed in the same system. Okay, so where do we go from here and what can we conclude? So I think LiDAR-based methods like these are as accurate in many cases as the GPM and other field-based methods, which is great. So it holds promise for estimating biomass at large spatial scales. It is logistically challenging. It still requires field ground truthing, and it is expensive, but it has the advantage of providing significantly larger and more continuous coverage. And so as an example, in those CLE plots. The sort of best monitoring that we did in the field was 1,800 disk pasture meter measurements. We have 240,000 1 meter squared estimates of grass biomass. And in the broader landscape, something like 200 million pixels where we can estimate biomass. So you can answer sort of qualitatively different questions with this sort of grass biomass data. Like I said, trees continue to be an issue, and this is something I'd love to chat with folks about who are maybe interested in this. But peeling back this tree layer so we can get at the grass is something that we're still working to do in a way that's a little bit less coarse than just chopping it. So I'll just leave this slide up here, take a look at this, but I'm out of time, so thank you very much, and I look forward to talking to you soon.
Thanks very much, Tyler. Really incredible where you guys are going with this. And you might all remember Andrew started studying ants. So I see where you're going with this. Next up, we've got Jetty van der Voort. And I'm not sure whether you're on a 5 or 10 minutes. The schedule says 10, so you should know.
So hello. Today I will talk about vegetation patterning in mesic savannas. So let's dive into it with some observations. So here on the picture on the right-hand side, we see a natural boundary between a savanna and a grassland, which is already a form of spatial self-organization. And in the grassland, we also see these patches of savanna trees going on. It's a type of vegetation patterning. And the goal of our research is to discover the mechanisms behind this pattern formation, and also discover how it influences the ecosystem resilience. For this, we developed a new mesic savanna model. For mesic savannas, it's important to note that there's more water availability than in the dryland spectrum, so that savanna trees can thrive, but there's not enough water yet for forest trees to be there. We decided to create our own new model because we wanted to take into account the spatial effects from the beginning, because this is crucial when looking into vegetation patterns. So what are the key model mechanics? We model fire, and we do this explicitly by modeling fire traps, where younger savanna trees experience an aging delay due to the frequent fires that occur into the mesic savannas. We also model shades, so larger type of vegetation have a negative effect on the smaller type of vegetation, and the spatial effects come into play via the seed dispersal. So I'm a mathematician, I like equations, but in this presentation I will not go into much detail on the model itself, but feel free to contact me during the conference. For now it's important that it's a reaction diffusion model, and the key model variables are grass and savanna trees, where we have pooled the savanna trees into the early life stage savanna trees and the mature savanna trees. So what does the model produce? It produces patterns. So to clarify what we see here, I have a 300 meter by 500 meter savanna landscape, and I plotted the grass biomass density. And the green means a high biomass density, and the yellow means a lower biomass density. So what we see here is spots of grass. And if we change the parameter setting a bit, we can also create stripes or gaps. It's important to note, I didn't have place on the slide, but the savanna trees are in exactly the opposite phase of the grass. So where there's more grass, there's less savanna trees, and where there's less grass, there's the more savanna trees. Mathematically, we were able to show that fire traps are essential for these patterns to occur. So that's one of the driving patterns, one of the driving forces in our model for the patterns to occur. So what about the resilience? There's actually two explanations that our model can give on the effect of vegetation patterns on the resilience. It could be a sign of ecosystem resilience, or actually an early warning sign that a critical transition is about to take place. Let's start with a positive one. So what we did here is we compared the full spatial model with the mean field model, where spatial effects are omitted. And we see on the left-hand side, we see that there's no grass left. The grass biomass density is 0. However, in the full model, grass is still being supported by these vegetation patterns. So in this case, the tipping is evaded by the by the spatial interaction. However, it can also be different. So sometimes the pattern forming bifurcation doesn't lead to stable pattern solutions. So what we see here is in a mixed savanna state, patterns are starting to occur, but as time propagates, we see that certain modes keep growing, and they keep growing exponentially until at the end time it takes over almost the entire plane. So what we see here is the transition from a mixed savanna state until a complete grassland state. So here there's no trees left anymore. So in this case, the vegetation patterning acts as a transient from one state to the other, and it can be seen that if you see these in nature, that it's an early warning sign that tipping is going to happen. So what do we still want to do? We now found two possible explanations, but we don't know yet what will happen when. So we're still trying to figure that out in our model. And we also still want to look at the more clear boundary between grassland and savanna and look into the boundary dynamics there. And as a last step, we would like to add the forestry variable so that we can also go into the humid savanna section. Yeah, with that, I would like to end my presentation. I want to thank the organizers for letting me speak, and I want to thank you for your attention.
Thanks very much. You kept perfectly to time there. Next up on the program, we have Tuban. He's going to be talking to us about vegetation changes in the south of Kruger and neighboring areas. Thanks, Tuban.
Good day, everyone. I trust you are well. So today I will be presenting some of the work that we have done here in Kruger National Park and surrounding areas, whereby we are looking at understanding the spatio-temporal distribution of bush encroachment in protected and unprotected areas. So I'm not going to bore you too much with the slides. I'm going to try to talk as much as I can without reading there so much. So it has been noted that savanna rangeland are going through gradual changes from being your open grassland to predominantly areas with woody vegetation, right? So now, in line with this, these changes, the bush encroachment poses threat into the savanna rangeland. This threat not only affects the biodiversity, but also the productivity of this rangeland, right? Now, this impacting on the sustainability of the ecosystem, and this can vary from the location management practices that are being done, and not only limited to that, but there are factors such as climate change and environmental variability. So now, there's this growing rate of bush encroachment also affects the livelihood, the rural livelihood, wildlife, and conservation, both directly and indirectly. In such doing, those who are driving around the Kruger National Park, you will see that there are areas that are heavily encroached, which also affects your visibility in terms of game viewing, right? So now, moving into the key thing that I'm presenting here. There have been a call by a number of scholars to do more research in terms of understanding the deeper, to have a deeper understanding of how this extreme climate condition affects the rangeland, right, using remotely sensed data. So in this, if you can couple the two, you can predict the ecosystem threshold, right? So now, some studies in response to that, they've noted that some of these encroaching species tend to inhibit areas that are, the climatic conditions are more favorable towards them, right? Now, again, in the southern Africa, it has been hypothesized that under dry climate condition, there will be a high rate of encroachment, which increases in density and cover. This is just a hypothesis that have been done because we have mostly semi-arid rangeland. So through the advances, back in the day when you talk of remote sensing, people will complain about the storage capacity, the efficiencies. So now, through the advancement of AI and computerization, we are talking of a Google Earth engine, which is a cloud-based computer, which is more efficient. There's no limitation in terms of space, right? So in this, the GE has been incorporated with machine learning algorithms such as your random forest and your deep learning, right? Which is more suitable for mapping and monitoring vegetation, spatio-temporal variability metrics of the vegetation, which include not limited to this bush encroachment. Now, in light of this, what the study seeks to do, it seeks to assess the seasonal variation of the spatio-temporal distribution of this bush encroachment across this. So we have also, what we did is that we grouped them into two, which we have the dry and the wet season. So we are dealing with those particular two. So all this was done in Google Earth Engine using Lancet data sets. Now, the study was conducted in the lower part of the Kruger National Park and the adjacent community. When you move through the help of the Sand Park scientific community, they gave us the guards to go outside and talk to us where we can find communal rangelands. So in this, sorry about that. So here, what we did in the model, in the GE, we have loaded the digital elevation model. We put the Landsat dataset from Landsat 5 to 8, and then we are using a 10-year interval from 1992 to 2022, which is a 30-year period. Now, again, we have to include the bioclimatic data, which is freely available also. And also, we have to put the soil characteristics. Now, we went to the field and collected the data, and then the data was split into two. 70% was the training data, and then 30% was the testing data. So all this, so one also particular aspect that is very critical, we use the spectral bands and the vegetation indices. This included your NDVI, your CEVI, your normalized phenology index, your moisture index, and lastly was the green vegetation index. So now, we did the classification using random forest. So at the end, we had the seasonal fractional cover from 92, 2002, 2012, and 2022. In this, we had the accuracy assessment and the spatial coverage. Now, if we can move along to the results what shows us, it shows that when we started in 1992, here, during the dry season, we're sitting at 2.2% of the encroaching species in the southern part of the park and also in the adjacent communities. Then when you look at again in 2002, we're sitting at 3.8 during the dry season. Now, when you look both in the 1992 and 2000 wet season, there was a slight increase whereby we're sitting at 3.8 to 2%, right? So if you can look on the results here without reading here, you can see that there has been a seasonal fluctuation of the encroaching species. So with 2022 having the highest level of encroachment at 10%. These are based on the model that we have got received on this analysis. So now we can say that the discrepancies between the vegetation in the invasive. So another one thing before I talk to Corey, the terminology here, I'm doing remote sensing. So when I'm saying invasive, I'm referring to the encroaching species, not native, not alien species, but just to clarify that one. So when I'm saying invasive, I mean encroaching species. So please don't chop my head off on that one. So yes, as I repeated, so we find that there was a significant variation in the seasonality of both the encroaching species and the native species in both the protected and unprotected land. So another thing that was worth noting is that during the wet and dry season, you can easily discriminate between the encroaching and non-encroaching. The reason being, through literature, some of the study, when we were doing the study, they noted that some of the encroaching species, such as like the sickle bush, they tend to lose their leaves later stage compared to some of the native species. So of which also, when we came here to do the field data, we also noted the same thing, that when we came around April, May, some of the ecosystem area had their leaves. So the spectral signature helped us in terms of discriminating that, because also we included the phenological cover, the normalized phenological index. So it helps us to discriminate this. As you can look at here, the spectral extent during the wet season, it is more than during the dry season, because now we are dealing with a remotely sensed image and we are using a high resolution such as Landsat, unlike if you're going to use your LiDAR or use a drone, which is the resolution is at finer scale, you can easily depict. So here there will be maybe spectral confusion that will happen. So we acknowledge also that particular area. So in terms of visualization now, we have found that most of the encroachment since from 1992 was mostly based here. So the white part were the mask classes that we're not interested in, like your build-up areas and all those things. So we masked them out. So these are not clouds. So now, as you can see here, this was a picture of 1992, what we got, and then 1992 dry, 1992 wet. As you can see, there's a bit of spatial distribution in terms of the seasonality. As I noted that during the wet season, we tend to observe more of the encroaching species. So as I noted here, if you can look at 2012, and this also, it was in a line, the study that I just was reading, and I found that Stellenbosch and Harvard and Dr. Smith, I think, they said the question was that they asked, is fire really the answer to bush encroachment? So what we found out here in this study is that, yes, climate variability and climate change influences the spectral distribution of these encroaching species. But then I'm coming to the critical part of the results now. These are just the overall accuracy results. So we achieved from 76 to 86 accuracy assessment from the random forest results. Now, but I wanted to come to this part now where we are talking about the important variables of the results. So we have found that elevation in all the years from 1992 to 2022, elevation was a most important predictor. And this is in line with other studies because elevation influences the soil texture, soil type, and also temperature. So now, if you can look at here, we have bioclimatic 12 and bioclimatic 4. Bioclimatic 12 is the annual precipitation, and then bioclimatic 04 is the temperature seasonality. So these were the two factors in terms of the climatic variables that were important into predicting the spatial-temporal distribution of these encroaching species. Now, what does that tell us? The results are many in line that to say that in the Savanna rangelands, in the southern Africa, temperature and precipitation plays a vital role in the spatial distribution of these encroaching species. When you also move into these indices, as I mentioned earlier, that the phenology also, the normalized phenological index also plays a critical role in giving us those high accuracy results that we saw there. They are not as high as Paul's results. I saw Paul achieve it, I think, 95 or 97. So they're not as high as his. I want to talk to him and see what he did and to get those high accuracy results. So now, when you look in terms of also the spectral bands, this also helped us in terms of achieving these results that we did, because now the spectral bands help us into achieving the separability between different vegetation. So now, so this is just also the same thing that I was talking about, that elevation also plays a critical role in that. So in closing, what we'll recommend, and never mind about that, I'm going to come that, is that as much as seasonality plays a role in the long, when you monitor it at the long term in terms of understanding how the spatial distribution occurs and in terms of elevation. There's a call that I was reading in terms of the paper that I was saying, if you have time, you can read it. It says, is fire really the answer to bush encroachment? They were looking at a 10-year period here in Kruger National Park. What I will also recommend is that we should put some of the drivers of bush encroachment into the model, such as fire and the grazing management, the grazing pattern, and see if we're going to see the same pattern in terms of the spatial distribution of these encroaching species. Now, the results demonstrate that in this study it's acceptable that it reached focus of the regional level, because most of these studies, if you can just do a literature search, you will see that it has been done at a larger scale using Lancet. Few studies have looked at it at the regional level. Thank you.
Thanks very much, Taban. We do have a few minutes left for questions.
Yeah, sorry, I forgot to acknowledge St. Parks and NRF for funding. Sorry, I just cut.
Not a problem. Any questions from the audience? Yeah.
Thank you. Very nice talk as well. I was wondering if you saw a big difference between the communities outside and the protected areas, if there was a difference in bush encroachment.
Yes, there was a difference, and then this difference, this is through the field observation, not to the results. The results, yes, there is a lot of difference. Okay, so pitches moved. If you can look at, as time move about, the areas that were masked out tend to increase in size because this will be your built-up areas and everything. But when we're doing the study, we realized that they are harvesting certain key of the encroaching species, like your Dichosistin area. They are using it for medicinal plant. When we're there doing the study, they are harvesting them. And now here, also in the Kruger National Park, we have noticed that some of the heavy encroachment was towards the Pretoriascope side, and mostly towards the northern side, as if like we are exiting towards the Kruger Gate. But then when you go towards your, what is that, Lower Sabi and, what is that picnic area? I forgot the area that side. Chokwane, yes, Chokwane. Yes, so the encroachment is at a moderate rate compared towards when you move towards the Skukuza area to the gate, which is mostly closer to there, and also the Pretoriascope. Thank you.
All right, Tabang, I think you're off the hook. Next up for our last talk before lunch is another familiar face at this meeting. Christian, over to you.
Yes, thank you very much, Ben. Great pleasure to be back here again. And I know we are 10 minutes late, but I cannot shorten my talk by 10 minutes. So sorry, I need my 15 minutes. But I think the results may change some of your lives. A long title and no authors, because many people are related and supported this work. What my talk is really about is the spatio-temporal monitoring capabilities, and it fits nicely to the predecessing talk because we are working with the Sentinel data. So this SunParks project, ColdEMS, is in a bigger context of a South African and German program of cooperation, the SPACES program and the SALDI project in our case, where we explored the Sentinel-1, which is radar data, jointly with Sentinel-2 optical data for land degradation features defined here by a publication by Graham van Maltitz and others. So behind this is a SALDI workflow I will show to you later. I move on quickly just to give you an impression of the European satellites flying. But important is this Copernicus program with all the Sentinels, the first one starting in 2015, becoming operational, let's say, 2018. So this is a new era. It's only, 6-7 years now of operational satellites and optical and radar domain. So now comes my 1-minute remote sensing lecture to everybody, or maybe just to keep you updated again on optical data, obviously coming through those transmission windows here in the atmosphere, coming from the sun. Looking at these optical reflectances like Landsat data or the Sentinel-2, which is the optical satellite, we are looking at the reflectances. And I just want to point out here, of course, thermal emission, that's what Landsat is doing, is a nice, emission is always nice to look to water. And I'm saying this because now turning to radar, we are in a very different wavelength domain, very long wavelengths. But there's actually also emission, there's natural emission, and not many people know that, in the microwave domain. And I'm showing you very exciting results on these natural microwave emissions for soil moisture. But we are using, of course, the Sentinel-1, the active radar, and looking at active radar, we are seeing very different things than the optical domain. We are looking at backscattering effects in the vegetation. So radar is much more related to the structure of vegetation. And as your microwave in your kitchen, what we see is water. So it's the structure, but it's the water content of the vegetation. So it's really different things we are looking at. Okay, so this was the 1 minute, I hope, remote sensing introduction. And now looking at the workflow, we have products that we derive from radar, we have products we derive from the optical data, and the idea is to come up with synergetic products. And this is where we are right now, really. It's all developing, and I hope many of you are interested in cooperation, or you're doing it yourself, because we have on 6 sites, and 4 of them belong to SunParks regions. We have data clusters, so these sites are 100 by 100 kilometers. and we are exploiting them with the repository, a GitHub repository of Jupyter Notebooks. And the fourth speaker mentioned Google Earth Engine, but there's another wonderful opportunity for you here. It's the Digital Earth Africa. Actually, our data clusters come here from Pretoria, from Digital Earth Africa, and they work with Jupyter Notebooks. So you can explore, you can access without any costs. You can access these wonderful data cluster and processing techniques. They have a full set of processing techniques. Now, I'm showing this animation. This is just a vegetation index. Kukuza is up here. Just to make you aware of what a data cluster means. It's spatio-temporal monitoring. That is not easy. Just looking here at the vegetation index, you could use all other products. The dynamic in the Savanna system is just absolutely amazing, and that makes it so wonderful and difficult to work with remote sensing. So a lot of variation that we have to come up. And now I said it's an update, my talk. So just 3 slides on last year's presentations where we looked, for example, the Mighty Time series, here starting in January 15, radar backscatter, and something, the coherence. It's a product, it's also an operational product. You can download it from Alaska SAR facility, no costs. And what this coherence tells you is also something about the stability. Now think about this, the stability of your vegetation structure. Yeah, this is now not related to water, it's about the stability and the volume of your structure. And what is so nice here, some textbook-like features, the backscatter goes up, the coherence goes down, and vice versa. And the differences between years of your maxima or the width of this amplitude tells you something about the ecological conditions. And Sentinel-1, the radar, is always in addition to optical. So this was one result last year. Another one was about producing woody cover and then having woody cover for 1 year here for the park, looking in change. And we have a poster out there, continuing work of our PhD student, Marco Wolcher, so please talk to him. And the third one was on surface moisture monitoring. And this is we have a surface moisture index that we retrieve from Sentinel-1, from the radar data. This is the southern border of the Kruger National Park. And you see all this variation showing even local precipitation events because radar is so sensitive to precipitation. And we have continued to work on this. Now the results, so the update for this year. These are our 6 sites again, and just 2 examples here from Mokala National Park and from Kruger National Park. The first one is on vegetation. And we are actually using, as I'm mentioning here, one author, it's actually a master student. We are using these data clusters in our master seminars. And he has been looking in Mokala using the South African National Land Cover from 2020, and now looking into change in these per land cover type. So this was now looking into this small subsite. These are the land cover classes that you can read here. And this is now the trend over a 5-year period of the EVI, an enhanced vegetation index, which is more sensitive actually than the NDVI. I would recommend that, like Venter did it in his famous web portal, also from Landsat data, EVI. So anyways, we see there's over the 5-year period there are some changes, and that's of course another exciting thing about data cubes. You can produce maps of change, so you see in color what has happened over that certain time frame. Now looking at the temporal variations over those 5 years, 18, 19, 20, 21, 22, going into 23, with the 3 land cover types, grassland, shrublands, and woody land, you see the EVI is sensitive, and you see marvelously the changes through this period with the increase of rain. Now talking about rain, another fantastic product, the MSWAP, the ensemble wet precipitation data, that's what it stands for. It's by all meteorological organizations. It's an 0.1 degree, 3-hourly product. It's the best precipitation product you can get on a spatial coverage. And so we compare it to these MSWAP data, and of course you see how the EVI reacts. Now, and this is our surface moisture index from radar, and you see some similar features, how the land cover types separate, and also really, this is so many stories behind these temporal signatures. What changes here, for example, with the grassland with an increase of precipitation then afterwards in a dry area. But this is for ecologists to do their work on this. Now looking again, what can we do with a data cube? We can look at trends of the maxima or the change of the phenological season on the length. And again, it's so straightforward. If you then compare it to precipitation, you're actually proving that impact, what the ecologists know, the impact of precipitation on grassland, but you can plot it, and you can plot it as maps as well. You see how important precipitation is to your EVI maximum or the length of the season. So you get all this relation of mapping and time series. Now, again, this is now the plot for the Mokala National Park, or parts of it, the trends of EVI. But we wanted to see what's the synergy with radar. And again, if you look at the EVI versus now the radar backscatter, we see that coming to higher backscatter and also higher EVIs, this cloud is falling apart, which actually means, okay, one thing is you're losing correlation, but losing correlation always means there is additional information. Yeah, something is hidden in here. And then what we did, that's more for the fancy image processing people here, we are looking at radar indices over those 5 years. We have been producing a principal component analysis. We are only using the first principal components, and then you put this in a clustering algorithm. But the exciting thing, this is the western part of Mokala, is here we forced the algorithm to 2 classes, and this is to 20 classes. Now, and you see that although these are statistical measures, that there is a spatial context. It's not an arbitrary map. Yeah, there are distinctive features in this. It's just grassland, of these grassland types related to the structure and the water content. So we are very excited about this map, and this is now one result for the vegetation, that we can go deeper into grassland monitoring and grassland mapping, actually, by combining optical with radar. Now going to Kruger National Park, and we have deployed, in each of the sites we have an in situ soil moisture measurement with a logger, and then 10 meters apart our sensors, so 8 sensors. And this is really thanks to Tertia Stridham. Without her, this work would not be possible. So this is the location south of Lower Sabi, and now we come to the surface moisture mapping. And what you see here is the only thing you should please remember is this black rectangle, and this is related to SMAP. It's a NASA satellite, Soil Moisture Active Passive it's called. SMAP, it's running since a couple of years, but the operational products are coming out only now. And I remember last year IGAS at a remote sensing conference, exciting results on SMAP, and I will show you what's so exciting about it. Now, we will compare the soil moisture information from this location with our SURFME index. Okay, but first let's have a look at these. Since January 19, this was deployed. We had some problems and gaps, but it's a very nice, very interesting time series. See the poster by Jussi Bade on all our soil moisture measurements in all 6 sites, please. Okay, so now here we have the MSWAP again, the precipitation. Here the black line is this in situ line from Lower Sabi. The green dots is the EVI, so similar to NDVI, but I think a bit better for vegetation monitoring. And now the red thing is that Sentinel-1 surface moisture. And of course, yeah, it runs back and forth. It reacts to each of the precipitation. It has some interesting here, like the minima go along here with a line of EVI. So there's some interesting stuff in here. Also, long stories and many stories to be told. But I have only 2 minutes left, and you want lunch. So now, and this is one of the results I think that maybe change the lives of some of you, because now what we see is the same thing. The black line is in situ, here the precipitation, green is the EVI, this vegetation index, and now look at this blue line here, how it follows our in situ data. I mean, this is giving me goosebumps, really, seeing how a passive microwave product, this is from NASA's map, follows a local soil moisture measurement, which is, of course, because it's passive microwave, it reacts very sensitively to the moisture that is in that large amount of soil. So, but this gives us now the opportunity to separate, having these nice correlations against SMAP with our in situ. This is our SURF-ME. So what this thing sees is the precipitation, is the vegetation structure. It's a mixture because it's active radar from Sentinel-1, and this is passive, and it very nicely correlates with in situ. But we couldn't prove this without the in situ station. Yeah, that's what was really necessary here in Kruger Park. Okay, so which means we can now separate, and this is the work starting now, between really the changing soil moisture in the ground and development of the vegetation structure on top. Okay, and this is all enabled through data cubes, and I hope that, well, please contact us or contact directly Digital Earth Africa. We have in all our 6 sites many cooperation partners, but we're really looking for more direct cooperation, exploiting our data cubes, using our Jupyter notebooks. And, well, big thank you to, of course, the scientific service staff, the section rangers, and all the game guards, and to our own ministry for funding our research. Thank you very much.
Thanks very much, Christian, for you kept perfectly to time there, and everyone's very hungry. Just one very important notice before you all go. If you're hungry and you want to eat, please make sure you're wearing your name tag. If you don't, they'll make you pay. And then, yes, just a big thanks to everyone who spoke in the session. It was a really great set of talks. Thanks all. And the other last thing, please be back promptly at 2 o'clock to start the next session.